So Melanie Marota from Merck. It's my pleasure to present here today. I have enjoyed the discussion so far this morning. So just a brief outline on the talk. I'll give a little bit of background on amorphous dispersions. I think folks have heard this a little bit, so I'll touch on it just quite quickly. Some overviews on QC dissolution, the impact of F2 failure in the regulatory space, and finally the conclusions and lessons learned. So enabling exposures, I don't have to tell this audience, but we have a lot of these poorly soluble compounds, the BCS class 2s and 4s. They make up a significant portion of our pipeline at Merck as well. So they represent a huge challenge when it comes to exposures. The drug is generally considered soluble when you can take the highest dose and solubilize it over 250 mils across aqueous media and across a variety of pH ranges. If we take compound A, and it requires 20,000 milliliters of water to dissolve a 100 mg dose based off the crystalline solubility, you have a considerable problem here. So a colleague of mine actually cartooned this up, and I loved it. It's like having to drink 40 pints of beer in order to dissolve that drug, or the equivalent of 5 stomachs. So essentially, we're not able to do that. I don't think most people want to take their drugs with 40 pints of beer. So amorphous solubility tends to be much higher, but that form is not as stable, and as we've talked about before, the inclusion of a polymer is required to stabilize it into that state. And then, you generally have sustained in vitro supersaturation, looking at somewhere around 50 mg per mil for this particular compound, and the conversion of the majority of the dose into that amorphous state to maximize absorption. Okay, so when we look at dosage form development considerations, we basically have this interplay of improved stabilization of the formulation versus desired dosage form performance. And I think, Dr. Prudhomme touched on this as well, right? You have this idea of patient-centric dosing where you want to make sure that the image that you're creating is sufficient for an individual to intake. So having, these low drug-loaded formulations that require multiple units are problematic, but they also lead to a much more robust dosage form. So you have to kind of balance the two. So for drugs that are dispersed in the polymer carrier, you have a lot of polymer carrier, and it's necessary. So you have this interaction between the drug carrier, you have anti-plasticization, you have drug molecular mobility because you want to induce stabilization over a long period of time, especially at these aggressive environments as well, and you have nucleation energy augmentation as well. So the carrier level has to be selected so that it can carefully balance those considerations with the desired amorphous state against having too much carrier where you don't get a sufficient drug load so that you have an appropriate tablet formulation. So increasing carrier levels actually limits the dispersion loading in the final dosage form itself. So that impacts image size and patient compliance, as I touched upon. But one of the other considerations is too much polymer can actually lead to gelling tendencies depending on the polymer loading and the type. And this can actually impact your overall tablet formulation, and especially things like your disintegration time. So I just wanted to go through the mechanism of dissolution. So for immediate release tablets, we typically look at a tablet formulation. We oftentimes can granulate it, and so you get this behavior where you can look at the granule disintegration, and then you can quickly dissolve that into primary particles. So the traditional mechanism for our tablets is that they just rapidly disintegrate into small particles, they then dissolve, and the dissolution rate is controlled by the dissolution of these small particles, right? So those are typically super fast, very easy to see within a few minutes within the dissolution apparatus. However, in solid dispersion formulations, especially when you have higher polymer loadings, you actually see that the dissolution is not governed by these primary particle dissolutions, but essentially by a larger particle, which is the erosion of the tablet, where you see the gelling of that matrix material, and then you have slow hydration of the tablet over a period of time until finally fully dissolved. So essentially, when you have this erosion type behavior, if you were to take that solid dosage form as it's going through dissolution and cut it in half at some point, when you take it out of the bath, cut in half, The interior of that tablet is still dry, only the exterior has been hydrated. So as a result, the tablets that slowly erode via that hydration layer and the flaking off of those granules into amorphous drugs, those are what's controlling the rate of dissolution for the overall tablet formulation. Now, dissolution tests, and this is specifically looking at QC dissolution as opposed to biorelevant dissolution. Typically in biorelevant dissolution, you've actually created, either you might be working at the solubility limit of the drug or above that solubility limit, and you're looking at creating sink conditions where you see the rapid dissolution effect. This is primarily looking at QC dissolution, where you're trying to get that amorphous dispersion formulation and a tablet form through a quality test so that you can then file it as a IR product. So there's 3 categories here. There's a single point, which is, you have a highly soluble drug, BCS class 1 and 3, and it rapidly dissolves. And the definition for rapidly dissolving is having 85% of your drug release in a QC dissolution within the first 15 minutes. There's a 2-point specification, which is what's actually the most routinely used for BCS class 2s and 4s, and they basically look at slow dissolving or poorly water soluble drugs where you just have 2-point specification around that 85% mark. And then the last one is dissolution profile comparison, where you have to say lot to lot, what is the product sameness between each of these different tests. So the goal would be, hey, it's rapidly dissolving. If I'm dissolving 85% in 15 minutes, I'm good. No worries there. If I need a 2-point specification, it's just saying these 2 points within a particular time frame are all within that 85% mark, and I'm also fine. The more challenging thing to do is actually to create profiles that overlay one another when you have to show that product sameness. And so in an erosion-based formulation, you end up being in the last category. And so when you look at QC dissolution profiles, the profile is performed under identical conditions for the product, but you have to do what's called an F2 calculation. And so you have to go, and our regulatory colleagues have a nice calculation for us to look at, but basically you're looking at a set number of time points, the dissolution value of the reference at those specific time points, and then the value at the test. So in this case, they tell you, you will know the similarity between 2 dissolution profiles based off of this greater than 50 value. So we can establish the sameness of 2 tablet formulations based off of this factor. And then they go on to tell you what you can use in terms of your dissolution time points. So the F2 standard is that you take 2 measurements that are considered less than 85% release and 1 measurement after 85% release for both products. And this is aligned between the EMA and the FDA. And then there's also Japan, where Japan actually states explicitly which time points need to be used. And they say it's an interval of 15 minutes, 30 minutes, and 45 minutes for PMDA. So if you want to have a global product, most folks just go with the Japan dissolution requirements because that helps you establish where you are, provided that the 15 and 30-minute time points are less than 85% release. And so this is just setting the stage for where we see the dissolution tests impact and coincide with, our ability to create this amorphous dispersion product that now has this erosion-based behavior. So this is when I was early on in my career. I developed this formulation. It had amazing bioavailability. We did a lot of development on this drug, and we tested various different amorphous formulations and found that this one worked really, really well. And QC dissolution wasn't really something I was thinking about too hard. And then we get to late stage, and we start talking about the sameness of the product. And I said, these are the dissolution profiles. And I look at them, and I'm like, they look the same to me. I don't know what the problem is. They said, you have a huge problem. You cannot show that your product is the same across the batch. So these are actually not all, these are all different lots of the same formulation made batch back to back to back. They tested a lot, and they said, they're not the same. And I was like, wait, we've been making this for like years now. Like they've been through clinical studies. We're at the end here. We're ready to file. What do you mean they're not the same? And they said that this wouldn't pass your registration stability studies. You wouldn't be able to claim that these 3 lots are the same and would therefore have the same bioperformance. And so we looked and we said, okay, if we look at the clinical supplies and we look at these registration batches, how could they differ? And so we looked at 3 lots of materials against a reference, and we're starting to see this F2 calculation. And you can see it's like almost there, but it's not quite there. They're 45, 47, 48. I'm like, well, is it really that bad? And they said, yeah, this wouldn't pass. This would mean that you wouldn't have a good lot, and this is just a subsection of the lots that you've made. So we went back and we retested other lots of materials, and we found these were not the same from a quality standard. Bioperformance was very stable throughout, but the quality of the product was not considered the same. So, I mean, we're like, oh, the tablet weights are within specification. The hardness of those tablets have very tight RCs. Like, the compression performance of these batches were all under tight controls. Why is this dissolution looking different? And when you look at the profiles beside the numbers, you go, they look very similar. Like, when you look at that bottom plot, you're telling me that these formulations are actually not considered the same. And it's an artifact of the fact that QC dissolution was based off of immediate release tablet formulations where drug dissolved from those primary particles. They weren't looking at these erosion-based formulations, and they certainly weren't considering that the amorphous solubility of the drug is what's driving bioperformance. It's not just the dissolution rate of the drug within these QC apparatuses. So what can we do? We can't change F2. That wasn't on the table as like, hey, well, maybe we can go back to the regulatory agencies and explain what's the impact here. We have to showcase it. So where is the variability coming from? Okay, when we look at this, we say, historically, where are the dissolution profiles? What things are above 85%? What aren't? So we looked at the time points before 30 minutes. And we see that the dissolution profile plateaus at 30 minutes and essentially plateaus at 100%. So within 30 minutes, you have full release of your drug. And that's happening across pH ranges. So this isn't a pH-dependent solubility. So if your drug is fully dissolved and released within 30 minutes, by the time it exits the stomach, you have readily dissolved drug available for absorption, so this isn't going to impact bioperformance. So if the F2 calculation requires these 15, 30, and 45-minute time points, can we see where the comparison breaks down? And essentially, of all the 3 time points, you can see that 15-minute time point, the 30 and the 45 are very tight. It's that 15-minute time point which causes the greatest amount of variability. Now, that 15-minute time point is based off of that erosion behavior. And so some of these tablets can dissolve fully in 15 minutes. Some of them will dissolve in 20 minutes. But that's not causing a biorelevant risk. That's causing a dissolution risk for us. And that's not something we can control because it's very stochastic how these can dissolve and break apart within that space. So the high risk of failure is due to that sensitivity at the earlier time points, which again may not be biorelevant. So how can we address it? So the first thing we think is, all right, let's just see if we can look through the process, narrow the processing space, and try to see if we can tightly control. We had controlled before, but maybe we can make it just a little bit tighter control in terms of hardness on dissolution. And we also considered, could we explore this through a PK relevant study and conduct a human bioavailability study? You can imagine that the goal was, hey, could you just tighten the process first before we go on this human PK study? Great that you have a thought process on this, but if that PK study doesn't show what you assume is true, that's going to be problematic for this compound that is literally on the doorstep of, can we file this now? So here we go through, and we look at dissolution of batches. So we took a batch, this is the same batch, and we just weight sorted them. And we looked at the difference between a low tablet weight and a high tablet weight and saw how that dissolution profile performed. And you can see the F2 here, right? So it's very sensitive to weight. So when we see this demonstrates an F2 sensitivity across one of our batches, where the disintegration time between these 3 weights, the difference of that was on the order of 3 minutes. 3 minutes. I have to control to a tightness of 3 minutes. So when I go to start a product manufacturer, I would test the tablets, and then over the course of each of those tests and in process, I have to make sure it doesn't vary by 3 minutes. That is not a commercializable process. That doesn't work for us. So this is really difficult. It's just showing to us as much as you'd like to control it, you can't even do it through an in-process test that says, now you're going to hit this, because over the course of that process, you're going to have these variabilities. So then we said, okay, let's look at the hardness. And so we did one of these overlays of the plot of percent RH as a result of hardness, and we said even on stability, if we were to hit a particular hardness, we know that the RH can kind of modify this a little bit, and the dissolution profile can shift as a result of the environmental RH. So then we looked and we said, well, if you needed to have exquisite control, what would that look like? Just theoretically. And we get to a 20.7 to 21.0 hardness range that would be acceptable enough across the pH range that we show and still meets this F2 criteria. So one thing we learned is moisture and hardness impact dissolution, but specifically, this is not tenable, right? You can't simply control the process to make sure you meet this F2 consideration. It's beyond the ability of our process controls. So this can't happen, so we know this is not a way, this isn't a lever for us to mitigate the F2 space. This doesn't mean for other products it can't be. We certainly should look at things like how we can control the humidity, the hardness, as well as the tablet weights to have that level of control. This product just fell woefully out of that space. So now we have to look at exploring a PK study. So we came up with a proposed design. So we did have a discussion early on that the dissolution profile is what we're seeking to show doesn't have a relevance on PK, and that it doesn't matter how you modify that adjustment, that dissolution profile. In this case, we decided to do it through the hardness of the tablet, but we very well could have taken one batch of tablets and then annealed them to different conditions, and then use the humidity to kind of modulate the dissolution as well. This was just easier for us to manufacture different hardness tablets. So we took the same material, all the same batch, so one common granulation, and we just compressed it at 3 different hardness values. So we chose those based off of different compaction stresses. So we have batch B, which is our fastest. That represented the lowest hardness we could possibly achieve for a tablet, but still had good friability. And it also represented a really fast 15-minute dissolution rate. So on that y-axis is that percent dissolve at that 15 where we saw the variability. So we see that 80% there. We see our target batch, which was what we'd pretty much been working with up until now. We didn't use our existing batch campaigns. We decided to do a brand new batch manufacture for the purposes of this. And then we made a slow batch where we made it at literally the upper limit of hardness or of compression stress that you could put to still maintain good tooling. So we were like, as hard as you could possibly hit this, let's hit it for this batch and make the hardest tablet we can and see how slow that dissolution profile is. And that dissolution profile gave a 30% release at 15 minutes. And you can, and I'll show you some of the profiles, but it sort of diminished all of the other time points as well. So we went with as fast as possible, as slow as possible, and what we felt we could do in general. And the red shaded area represents what we think would have been the bounded space by the F2 dissolution, which we were kind of falling out of regardless. So here we go. So really cool. We hit what we wanted to. We saw that the batches, when we made them, theoretically landed where we wanted to be. We did the F2 calculations. We're seeing, relative to that bio batch where we alerted it earlier, where these things fell. So the fast dissolution profile actually met F2 with our bio batch. But our target formulation and the slow all were not F2 comparable. So we do have 3 distinct profiles. And then when we compare them against each other, the 34 and the 39, we see that, again, they are distinctively different. These are failed batches against each other. So target A batch is the reference profile. That was the center of that pilot scale development. Batch B was the very rapid profile, low hardness, met friability. And batch C was the really, really strong tablet, whereas the plateau of the compression profile allowed us to hit the max force of the tooling. So this is just illustrating, that red curve is the fastest dissolution profile. The X bio batch was kind of the reference of where we're at. We decided to create a brand new reference, which was the batch A, and then we did batch C, which was the slowest profile. So when we designed the study, we looked at it as a 3-way crossover human PK study. So we had 24 healthy subjects per arm. We had already ran some statistical data sets based off of our other PK data from earlier clinical studies. And so N of 24 might seem like a low number for this type of a study, but it's because our PK was actually not really variable. I mean, this is a drug that just looked so good across all the clinical studies. So when they came back and they said the study power is 24, we're like, really? Because all these batches are having these very different F2 numbers. So it actually gave us confidence that, hey, this might actually work. This was meant to be a single dose, open label. The study objective was just to estimate the PK parameters for both AUC and Cmax, for which we felt were the relevant PK parameters to show efficacy of the drug against the test articles. So you can see 8 patients per arm, and then you can see the 3-way crossover as we went through. So I just wanted to state this isn't the first time somebody's looked at, QC dissolution and looked at exploring the relationship of that dissolution on PK. This was just one of many publications, but it looked at assessing this performance for dispersions in general across 40 different compounds where they sought to get IV-IVR. This is one of the things that I use to kind of sell the idea of doing these type of PK study to my management to say, listen, this isn't the first time this has been a problem. This is something we can use. It's a good tool for us. And in that case, they found that in the majority of compounds, they actually were able to see some sort of correlation between the dissolution and the PK. And so it's important to examine which factors affect dissolution and absorption in order for you to showcase, hey, this is going to be a good tool for you to use. But the application of something like this for IR products is just not very common because you don't have to do it. This is a particular space for dispersions where it becomes necessary. So we get our human PK results, and I'm very happy. So the AUC across the formulations gave GMR within 90%. It was all within the confidence intervals we wanted. The Cmax comparison was also within the confidence intervals we wanted, and we went to excellent boundary conditions. So from a process capability perspective, our initial bounds were really tough, like we started before. The hardness was very, very restricted. You had difficult time achieving the LC for 15 minutes dissolution, to when you can take the totality of that data, you now fell into an excellent space where you increased your process robustness. In trying to counteract this F2, we now widened the space of the hardness tablets we could get, which was great, and also widened the spec for the LC, which was the goal. And so you can see the data here. I mean, the PK profiles overlapped really exquisitely. It was a really nice test case for us to showcase the PK really didn't get affected by these types of extremes. And we went to the extremes. And there was some discussion of how extreme should you go, but there was a lot of confidence that this didn't have an effect on PK, as evidenced by other clinical data sets. So again, in conclusion, a clinical study can be used to expand the space for manufacturing that might otherwise be constrained by regulatory considerations. Bioavailability of poorly soluble drugs is enhanced through that amorphous drug conversion, which is further stabilized through the carrier polymer, but the carrier polymer may deleteriously impact your dosage form performance. So process controls can be used to sufficiently de-risk these regulatory equivalency requirements, but they may not always be feasible. So you can use a clinical study to serve at expanding that processing space. And that's it. And so I just want to thank my colleagues.
Thank you very much, Melanie.